everyone, welcome. This is Dr. Jeks, your science teacher. Today I'll be demonstrating the food tests which are necessary for you to pass your Cambridge IGCSE, Cambridge GCSE, your ZIMSEC O level, combined science and biology practicals. So here I'm going to show you a demonstration of the practicals for IGCSE, GCSE, combined science and biology, and ZIMSEC O level biology and ZIMSEC O level combined science. I'm going to start with the starch test. So with me here I have two food samples. We've got a potato disc and bread. So we're going to check whether potatoes or bread have got starch. What we'll need is some drops of iodine solution which we're going to put onto the food samples. And then any color change which we're going to note is either going to tell us it's going to help us detect whether they are or there is starch inside the food samples. So I'm going to add here 1 drop or maybe 2 of the iodine solution, and we're going to observe the color changes which take place on the potato disc. You can already see here that the tissues are turning darker, black. The iodine color, which is the iodine solution, which is brown in color, is slightly changing to a blue blackish color. So that indicates a positive test for the iodine test or the starch test. You can see here that our potato is turned black on the regions which we put the iodine solution, and this blue-black coloration is indicative of a positive test for starch. So the first test is done. We're going to look at the second sample, which is bread. I'm using here a piece of brown bread. Some people believe brown bread is healthy, so we're going to test again if the presence of starch or the presence of starch in our piece of bread. So I'm dropping here 1-2 drops on my piece of bread. You can see that it immediately turns black. It's an intense blue color which appears black. So this is indicative again of the strong presence of starch inside the bread sample. So this is a positive test, and it confirms that our bread does indeed have starch. We will now move on to the next test, which is the protein test using egg solution and milk solution. So I have 2 protein samples, my egg and my milk. So firstly, I'm going to make an egg solution. What I'll simply do, I'll break the egg and add it into some water in a beaker, and then I'll mix with water as well as the milk solution. I'm also going to use the milk to make milk solution. I'll simply add some water to the milk. The reason why we do this is so that we dilute them, because they are too concentrated, so we need to make them dilute. Here's my egg solution. I broke the egg and added some water, so it somewhat appears brown or yellow. And this is my milk solution. I've mixed my milk with some water, like I said, so that I can have the milk solution. Obviously, take note that you need to label your samples so that all your beakers do not get mixed up. For the protein test, I'm going to use sodium hydroxide first and then add copper sulfate. Here I'll take a clean test tube, add my egg solution, maybe just 1 cubic centimeter. It will be more preferable to use a syringe to measure out small volumes so as to get accurate measurements. But this is just a quantitative test for demonstration. Then again, I'll add a similar amount of sodium hydroxide. Observe the color changes which take place. As soon as I add the sodium hydroxide, it decolors the egg solution, becomes transparent and somewhat colorless. You can see the difference here. This is the egg solution, this is the egg solution plus sodium hydroxide. So it has changed. Again, here you need to use a dropper to add a few drops of copper sulfate. It is not advisable to just pour in the chemicals because you miss the end point.
So I'm going to use my dropper. I'm going to add a few drops of copper sulfate into this mixture now. I'll add my drops 1 and 3. Take note of the color changes which take place here. It starts with the blue of the copper sulfate, but immediately turns purple. So if you put 3 or 4 drops, it will be fine. You will observe that there's a purple coloration. This purple coloration is a positive test for the burette test, or the test for proteins in a food sample, after having added sodium hydroxide and a few drops of copper sulfate. So that test was positive. I'm going to repeat this test now, but now using a different food substance. In this case, I'll use milk. I've added my milk to some water, so it's a milk solution. I take another clean test tube. I'll add here my milk solution, maybe a cubic centimeter. Then I will need to add again my sodium hydroxide, maybe another cubic centimeter of sodium hydroxide. You can see that it becomes maybe a lighter shade of white, pale or off-white, if you can say. It's not that intense white coloration that was there. And then I'll add my few drops, maybe 3 or 4 drops of copper sulfate. 1, 2. You can see that the color is changed from white to slight bluish, and then that purple coloration is back again, although it's a bit more intense as compared to the one from the egg solution. You can see here that the egg solution is clearer, but the milk solution is a bit obscure, looks like a suspension. That's because of the fact that milk contains a lot of minerals like calcium, which do not dissolve very fast, in like the proteins which are present in the egg solution. So next I will do my fat test, or the emulsion test. I'm going to again take a clean test tube. So in the clean test tube, I will need to add my sample of oil. So in this case, I will use simple cooking oil from the kitchen. Here's my cooking oil. It's made from vegetables. So I'm going to add my cooking oil to the test tube. This normal general cooking oil from our test tube. Maybe add a cubic centimeter of cooking oil to our test tube. Next, I will add corresponding amount of ethanol. Then my ethanol, I've already put it in a test tube, so I'm using absolute ethanol. So you can see here that there are 2 different layers. The one at the bottom is the oil, the one at the top is the ethanol. This is because ethanol is less dense than oil, than cooking oil, and hence it is on the top. So these are called different phases of the liquid state. Okay, although both of them are liquids, they have different phases. One is less dense than the other, so it floats. The ethanol floats on top of the oil. You may have noticed this, that oil also floats on top of water. So now we're going to shake. We close the top of the test tube with our finger. Remember to wear gloves so that no chemicals gets onto your skin. So gloves for safety. So shake vigorously the mixture of the oil and the ethanol. This is so that the ethanol can break down the fats or immerse itself between the particles of the fats. So now you can see that we have the fats being broken down into small fat droplets. What we need to do now is to add the mixture into a clean test tube with water. So take a clean test tube, add some water to it. You take a clean test tube, add some water, some clean water to the test tube, just enough so that you can be able to follow through. Make it more than maybe about half, so that you can be able to see. Now I take my test tube, which I have the fat and ethanol mixture, which I have shaken, and I'm going to pour. I want you to notice what happens when I add this mixture. Just take the top part, don't add solids to the water. Take note, when I pour only the liquid part, an emulsion forms that emulsion is the positive test for fats. It shows that fats have been broken down and it forms somewhat a cloudy white on top. So that emulsion there is an indication that there are fats present in our sample. Alright, so that is an indication of a positive test. So what has happened here is that the ethanol is broken down the fats into smaller pieces, and these smaller pieces are the ones that form that cloud or that emulsion, and this is why it is called an emulsion test. Alright, so there we have our 3 tests which we did, and this marks the end of our food test. For an explanation of why these tests are happening like this, to explain these results, please watch our other video highlighted at the end of this video.